Assalamualaikum to our respected lecturers Senarzul Hadi. Today, me, Ehya, and my friends are going to present about skeletal muscle glutamine. So basically, what is glutamine? Glutamine is the most abundant free amino acid in the body. It produced in the muscles and is distributed by the blood to the organs that need it. It also important for providing fuel, which are nitrogen and carbon, to many different cells in the body. Other than that, glutamine is needed to make other chemicals in the body, such as other amino acid and glucose.
There are a number of enzymes that are involved in the glutamine metabolism. The two main intracellular enzymes are glutamine synthetase and phosphate-dependent glutaminase. With respect to the intracellular location, glutamine synthetase is primarily found in the cytosol, whereas phosphate-dependent glutaminase is mainly found within the mitochondria. These locations are compatible with the enzyme function. The function of glutamine synthetase is to produce glutamine for the synthesis of cytoplasmic protein and nucleotides. The function of phosphate-dependent glutaminase is to catalyze glutamine conversion to glutamate as an important step to the tricarboxylic acid cycle entry at 2-oxoglutarate as an energy source or source of metabolic intermediates.
Next, we are going to talk about glutamine synthesis and hydrolysis. The synthesis of glutamine mainly occurs in skeletal muscle. From glutamine, glutamate is produced through glutaminase activity, releasing ammonium ion. This reaction is called as glutamine hydrolysis. Glutamate is also synthesized from alpha-ketoglutarate and ammonia. Muscle cells can synthesize glutamine through glutamine synthetase using glutamate, ATP, and ammonia.
Glutamine-consuming tissue. Some types of cells, such as the immune system cells, kidneys, and intestine, show a higher use of glutamine that classified them as glutamine-consuming tissue. This is because these cells have high glutaminase (GLS) enzyme that increased glutamine hydrolysis. Glutamine synthesizing tissue. Skeletal muscles, lungs, livers, brain, and fat tissues have a higher capacity for synthesizing glutamine that classified them as glutamine synthesizing tissue. This is because these cells have high glutamine synthetase (GS) enzyme that increased glutamine synthesis. Liver is the tissue that exhibits the highest capacity for glutamine synthesis, but the main tissue for glutamine synthesis, storage, and release is skeletal muscle because it represents of approximately 40% of total body mass. However, this classification is depending on body homeostasis. This means that there are conditions that glutamine synthesis cannot meet the needs required by organism, which specifically during catabolic situations.
This section will be discussing about the importance and function of muscle glutamine store in health and catabolic situations. Skeletal muscle constantly release glutamine into the blood and feed many other cells and tissues such as the immune system, gut and liver through intertissue metabolic flux. So, during health, which is in normal condition, gut or gastrointestinal tract is the principal or major organ of glutamine uptake, while the main organs of glutamine synthesis and release is muscle and liver. During catabolic situation, skeletal muscle become the major glutamine exporter, which means it releases glutamine more than usual. The effect of this condition is plasma glutamine might not be sufficiently depleted in overtrained athletes or people involved in exhaustive exercises. Conversely, during this catabolic situation, the liver and immune system cells become the major sites of glutamine consumption. In this situation, there will be a reduce of glutamine uptake by the gut, so it can facilitate bacterial translocation into the blood and systemic inflammation. The example of catabolic situation are trauma, surgery, sepsis, and exhaustive exercises.